Hello everyone, I'm Dario Righelli and I'm a postdoc at the Department of Statistical Sciences at the University of Padua in Italy. Today I'm going to present epigenomic enrichment analysis using Bioconductor. The main aim is to compare more computational methods and provide guidelines on epigenomic data analysis. And to do so, we use three different datasets. The first one is a HiTSeq dataset with 4 biological replicates and 2 conditions. This dataset is used to catch differences in open chromatin regions. The second dataset is a ChIP-seq dataset of one condition for nine biological replicates. And we use this dataset as a control dataset to understand how many random differences are we able to catch. So a sort of false positive discovering. And the third one is a spiking HiTSeq dataset of 3 biological replicates and 3 conditions. And this is useful to understand how many known differences are we able to catch inside the dataset. So the main idea is to have, this is the main idea of the work. So we are using this Scan2, Max2 plus DiffBind and CISO software in order to construct a peak consensus and a matrix of this peak consensus versus the samples. And each cell of this matrix is represent the number of reads for that specific peak inside the sample. The main differences between this software is that this Scan2 and CISO have their own peak collars, while DiffBind needs to use external peaks from external peak collar. But this Scan2 has the possibility to work also with external peaks from external peak collars. So we choose Max2. The differences for the differential enrichment between the conditions are computed using EdgeR, but you can use other methodologies that you prefer. The first aspect that we investigated is the normalization of this kind of data. So applying different normalization methods for RNA-seq data, we obtain different numbers of differential accessible regions for the HiTSeq data. So this suggests that maybe a more specific designed normalization is required for this dataset, for this kind of data. Here with this upset plot, we are representing the differential accessible regions computed across the methods. So we have this Scan2, DiffBind with narrow peaks, DiffBind with broad peaks, and CISO. The good part is that all these methods are able to catch the same, the biggest number of differential accessible regions altogether. But when we go to investigate the single methods, so the dots, we can see that CISO has a very high number of DARs by itself, and the same happens with DiffBind. So this suggests that maybe these methodologies catch a high number of false positives. The same happens with this Scan2, but we have a lesser number here of false positives. With the ChIP-seq dataset, we have just one single condition. So in order to catch the differences, we permuted the samples 126 times. So because you can have just 126 permutation with 9 samples, and we randomly divided into groups, and then we applied EdgeR, and as you can see, this is a box plot, and here you can see from CISO that CISO catches a very high number of differential enriched regions here. So we have 90,000 in terms of number of DERs. So here you can see the same plot without CSO, just diff bind and this scan2 with and without normalizations. And in this case, you can see that, of course, we catch false positives, but in this case, they are less. And this is the group who is leading this project, and I want to thank them all and all of you for your attention. Thank you.